This is Mr. Maxwell for Pingree AP Biology. I gave you guys this sequence here, which you should notice has U's in it and therefore RNA sequences. And then I had you copy and paste it into NCBI BLAST. And what you should have discovered when you did this was that it turns out that this is the entire genome for tobacco mosaic virus. I don't really care so much that you learned about tobacco mosaic virus, but more important thing here is that you learn how to use these tools. This unit that we're doing is centered around information, and there's a whole field of biology, new field, called bioinformatics, which looks at biological information. And so these tools are all available online, and I want to give you some familiarity with them because we'll be using them. Back to TMV, it turns out that tobacco mosaic virus encodes 6 polypeptides, and I had you guys look at each of the 6 and figure out how long each gene was and what it did. And when you add them all up, it turns out that you should get more information than is available in 6,191 bases. In other words, you need more space than the genome actually has to encode all those genes. And the way the virus does this is that it uses overlapping sequences in order to condense the information. And you can see this is the length of the genome of the virus, but it gets several different things out of the same sequence. Okay, so a little bit about viruses. It turns out the tobacco mosaic virus was the first virus discovered. Its genome is about 7 kb long, 7,000 bases long. So it's on the low end of viruses' genomes. They range about 2 orders of magnitude here. And then bacteria have genomes that are about 10 to 100 times bigger than that. And then eukaryotes all have larger genomes yet, with us being around 3 gigabases or 3 billion nucleotides. Graph's nice, it summarizes everything, but I have a hard time really getting my head around what exactly that means. And so if you think that tobacco mosaic virus takes up the front and back of a page of paper, an E. coli genome is about 1,000 sheets. So 500 sheets of paper in a ream. This 2 reams would be 1,000 sheets, so front and back of these, and you have enough information to get you to program an E. coli cell, a bacterial cell. To program a human cell, you need 2 of these flats here. Each one of these flats has 120 boxes on it, and each box has 12. Each one of these boxes has 12 reams of paper. Okay, so it takes 2 whole flats here to make up the human genome. So, much larger, of course, but that's the range of what we do. So let's go back to our friend tobacco mosaic virus and take a look at the symptoms. This is a tobacco plant, and here you can see this yellowing is called chlorosis, and then this dead patch is called necrosis. And so the virus causes chlorosis and necrosis on the plant leaf, and of course that reduces the surface area the plant has to do photosynthesis, as well as in replicating the virus, the tobacco plant is no longer using the energy for its own needs. And remember, the last unit was energy, so here's your connection between energy and information, or at least a connection. So this is an electron micrograph of the virion, the virus particle. A lot of people will use the term virus and mean the virion. Technically that's not correct, but for your level of AP Bio, you'll see people do that, you'll see books do that, and I wouldn't worry about it. But now you know better. This is actually a virion, not a virus. It's a virus particle. This is the little package by which the virus moves between organisms. Okay, so it's about 18 nanometers wide and about 300 from end to end. And just to give you a little scale, remember that red light is about 700 nanometers in wavelength, and so this virus, this virion, is about half of the size of a wavelength of red light. So hence, you can't see it with light. This is why you have to use electrons to bounce off of the virion so that you can visualize it. Now, it's true that recently um, uh, some really smart people have figured out how to get past the the wavelength limit on light microscopy using interference patterns and everything, and that's really cool. But the point is, the thing's really, really small, and so there you go. So uh, it's a message sense RNA virus, which means that when it gets in the cell, it looks to the cell like a messenger RNA. And so the messenger RNA, a messenger RNA would go to a ribosome and get translated into protein. And that's exactly what this virus does. It looks to the host cell like an mRNA. And so the host cell ribosome takes the mRNA, which is the virus, and translates it into an RNA-dependent RNA polymerase. So the first three parts of this open reading frame here, plus this little open reading frame 2, the virus gets 3 proteins out of that, which compose an RNA-dependent RNA polymerase. An RNA-dependent RNA polymerase reads RNA and makes RNA. So one of the things that the RNA-dependent RNA polymerase does is reads this RNA and makes the opposite strand, which is complementary to it. And once it's got that complementary strand, then it can make more plus sense virus. In other words, plus sense virus RNA is also going to be message sense, and is going to make more RNA-dependent RNA polymerase. Okay, so we got that going on. The next thing that we have, once we've replicated, is now that we have the minus sense RNA, and some of those minus sense RNAs are short RNAs here, subgenomic RNAs. This open reading frame 3 is actually on the minus sense strand. So it's not even available to be copied when the virus first gets into the cell. It has to be it's not in plus sense. It's on the opposite sense strand. So once you've replicated this RNA and copied it into its complementary strand, now the movement protein's in the right frame, in the right sense to be able to be translated. Okay, so once you have the subgenomic RNA, then you can make your movement protein, which allows the virus to move cell to cell in the plant. So we have replication, then we have spread, cell to cell, and then when we replicate this strand, we can make a subgenomic RNA from it, which is now back in the plus sense, and that is what makes the coat protein. And the coat protein is what surrounds the viral RNA and allows the viral RNA to move from organism to organism. Okay, so from plant to plant. Okay, so let's go back to how do you get 3 pieces of protein out of this particular stretch of RNA? Well, what you do is there's a start codon over here, and you can translate 1 polypeptide. And sometimes it'll truncate, and if it truncates, that's fine. You get 1 polypeptide made from here. Sometimes, though, when the ribosome gets to this spot in the RNA, it hits a thing called a pseudoknot. This is basically the RNA coming back and folding on itself and binding to itself in particular complementary regions here. And so the ribosome gets here and it stalls. And when it stalls, there are two ways in which this can happen. Um, but let's just suffice it to say that the ribosome slips a little bit, and that puts it in a different frame and allows it to disrupt this pseudoknot and then do what's called a read-through. So basically, with the read-through, it allows you to get one polypeptide that's full length, another polypeptide that is just this one small unit, and then a third polypeptide that is just this open reading frame 2. So you get 3 polypeptides out of that one sequence. Really cool way to kind of condense information and use all the space that you have. Okay, so this is kind of an overview of tobacco mosaic virus's strategy. The genome is positive sense. It gets inside the cell. Once you've made your polymerase, your RNA-dependent RNA polymerase, you can make the minus strand, and then you can make the more of the genome sense. Then that gets translated, and traduction is actually the French for translation. So this image came from a French paper, but just remember this is translation actually, and that translates into a bunch of proteins. Okay, now so how do you move cell to cell? Well, if you remember that plants have these little connections called plasmodesmata, and it turns out that plasmodesmata actually have endoplasmic reticulum that is continuous through the plasmodesmata. And what the movement protein of tobacco mosaic virus does is it rides on the endoplasmic reticulum and grabs onto the viral RNA and allows that to transport through the plasmodesmata and then get into a second cell. So it uses this highway that plants have to move RNAs from one cell to another, or to move materials from one cell to another, to move its RNA. Pretty clever. And there are other ways by which other viruses do that, but I just want to focus on our tobacco mosaic virus. It's a monopartite movement protein. This, again, is the virion, and it turns out that the virion is made up of multiple copies of the coat protein. And so, each one of these units here that are shown in darker blue and lighter blue are the same actual protein, but they're just showing them so that you can see where one ends and one begins. And then this red here in the middle is the RNA. So you can see it coats it pretty well. And here's another view of that. This is the crystal structure of the coat protein assembled without any nucleic acid in it, without any RNA in it. And then you can see where the RNA is in this picture here. And you can see that it very nicely packages the RNA and makes it so that it's very stable outside of the cell. In fact, at the University of Wisconsin, there's a jar of purified tobacco mosaic virus from back in I think the 20s or the 40s when this was first purified, that was still infectious, I don't know, 15 years ago when I was there. And that's it. That's all you need to know about that. Hope that helps.